Nitrosamines are potent genotoxins and probable human carcinogens. Some common examples are NDMA and nitrosodimethylamine and NDEA and nitrosodiethylamine. Nitrosamine contamination recently emerged as a global regulatory issue after the US Food and Drug Administration has found some nitrosamine impurities in one product on the market. It was valsartan in 2018, and this is a commonly used medicine for high blood pressure. Also, FDA has found some nitrosamine contaminations in other drugs products on the market, such as ranitidine, that is a medicine for the heartburn, as well as metformin, which is a drug for treating diabetes type 2. On the other hand side, on European market, European Medicines Agency, EMA, has also initiated some risk assessment activities for the drugs that are on their market. They have issued guidance for drug manufacturers how to avoid and reduce nitrosamine contamination in their drug products. To solve this issue and protect patients' health, drug manufacturers have now legal obligations to do a thorough risk assessment of their products on the market. If necessary, they will need to implement some mitigation strategies. Some of them might include reformulations. If they do not do that, they risk that their drug products will be withdrawn from the market. The prerequisites of nitrosamine formation are, on the one hand side, the presence of functional groups that are susceptible to nitrosamine formation. Those are, for example, secondary amines, which are vulnerable amines. On the other hand, the presence or in situ forming of nitrosating agents that are easily formed from nitrate. So in the first activation step, nitrate can easily transform into the nitrosating agent, especially under acidic conditions. In the second activation step, those nitrosating agents can react to vulnerable amines and the nitrosamines are formed. Multiple sources of nitrosamine contamination are possible. Nitrate is often present as an impurity in excipients, whereas secondary amines can be part of a structure of drug substance or its impurity. Also, a source of contamination can be a buffer, a solvent, or something during the cross-contamination during the production line or during the production of raw material. So there's a wide variety of conditions when nitrosamines can be formed. However, they are most likely formed under acidic conditions. At risk are drug products that contain drug substances whose structure comprises secondary or tertiary amines or tertiary ammonium salts. At risk can also be drug products whose drug substances are stabilized using buffers with tertiary or quaternary salts. However, at risk can be also drug products whose excipients has structures that comprises secondary or tertiary amines or also quaternary ammonium salts. A well-known antioxidant that can be used as nitrosamine formation inhibitor is ascorbic acid. Another well-established antioxidant that is often used in food industry is DL-alpha-tocopherol. It can also be used during the formulation drug product development as nitrosamine formation inhibitor. The option of adding either ascorbic acid or DL-alpha-tocopherol to drug products is offering formulation scientists worldwide a new and safe opportunity to redesign their pharmaceuticals, as well as at the same time to keep the nitrosamine formation at low level. DSM has developed its expertise on vitamins specific in ascorbic acid and DL-alpha-tocopherol over the last 70 years. Today we have the largest portfolio of vitamins both in food and pharmaceutical grade. DSM has expertise in formulation, quality and regulatory that can help drug formulators worldwide to overcome this urgent regulatory issue in the pharmaceutical market. DSM has a variety of ascorbic acid products to offer. For example, they can be used as excipients, but they can also be sold and used as APIs. Those grades are offered with full regulatory documentation and are produced under pharma GMP standards. DSM has also several grades of DL-alpha-tocopherol that could be used for pharmaceutical usage.